Greetings. I'm Yvonne, Stat for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Today, we discuss one of the major concerns about climate change, the impact of ocean acidification on marine life. Our guest is Dr. Louise Cameron, a marine ecologist and carbonate chemist, presently a postdoc investigator at Woods Hole Oceanographic Institution, Massachusetts. Dr. Cameron joins us to explain ocean acidification itself, and especially its effect on vulnerable marine bivalves like scallops. We'll also learn how marine scientists investigate the effects of ocean acidification. We're very pleased to welcome Dr. Louise Cameron. Thank you for joining us.
Hello, Yvonne. Thanks so much for the introduction. Thank you so much for having me as well. It's great to be here.
Right. We really need this information. And so I'm going to ask you from the start here, would you please explain what ocean acidification is and what causes that?
Yeah, of course. Yeah. So as we're pretty much all aware of, global warming occurs when we release fossil fuels into our atmosphere. And the reason why this happens is because these fossil fuels act as a kind of a blanket, and they trap in long wave radiation that is normally reflected back off the Earth and goes back out of our atmosphere. But not all of the greenhouse gases we produce stay in our atmosphere. In fact, around 50% of all anthropogenically produced greenhouse gases are dissolved in our oceans. And that is a good thing, because if they didn't dissolve in our oceans, our planet would be warming at a much faster rate than it currently is. But some things happen when they dissolve in the ocean, in particular carbon dioxide, which is the most prominent greenhouse gas that we produce. When it dissolves in our surface oceans, it undergoes some chemical reactions. And these chemical reactions eventually produce carbonic acid. And this lowers the pH of seawater, and it also lowers the amount of carbonate in our seawater. Now, this is really important for a lot of marine organisms, because carbonate is the building block that animals use to build their shells and skeletons. So anything that has a shell, like a bivalve, or a calcium carbonate skeleton, like a coral, could be affected by ocean acidification.
And that's why we're seeing so much destruction and so rapidly. Thank you for that part. Now, is there a different effect depending on the depth of the seawater or the particular oceans involved? What are we talking about there?
Yeah, that's a really good question. So I talked about how carbonate declines in our oceans under ocean acidification, and the concentration of carbonate is known as the carbonate saturation state. This is a kind of kinetic property. So when saturation state is higher than 1, we say that that water is oversaturated or saturated. When it drops below 1, we say that this water is undersaturated, and that is kind of the threshold that causes dissolution. So below a saturation state of 1, we would expect shells to naturally dissolve in seawater. And this saturation threshold can be affected by a number of different things. So one thing is depth. The saturation state gets lower under pressure. So in deeper waters, saturation state is naturally lower. But what we call the saturation horizon, which is the depth at which water becomes undersaturated, is shifting because of ocean acidification. So it's getting shallower. So more waters are becoming undersaturated. Carbon dioxide is also more soluble in colder waters. So, for example, our poles are acidifying faster than our tropical regions. So yeah, high latitude regions tend to be pretty vulnerable to ocean acidification. Another thing that can make an effect is other sources of acidification. So ocean acidification is the long-term effect of greenhouse gases, but we also talk about coastal acidification, which is acidification that's caused by runoff of nitrogen and organic carbon from farming, for example. So this activity causes algal blooms in our coasts. When these blooms die, they decompose and release carbon dioxide. So that's kind of a rapid acidification that happens in our coasts. And this can get exacerbated by the other long-term process of ocean acidification going on in the background as well.
Oh, that is interesting information.
Yeah.
So this is, and since it apparently impacts coastal regions,
Hmm.
then does that make the link with shellfish the more dramatic because they're not usually in the deep sea?
Exactly. Yeah. Yeah. The shellfish that we eat are all in coastal or shelf waters, which are more vulnerable to acidification than more open ocean stable conditions.
Okay.
Yeah.
Now, what's happening to those, just the bivalves, the shellfish in general, then, since they're located more in
Hmm.
coastal regions? What's
Yeah.
happening to those populations?
Yeah, so it depends a lot on the species. So another thing that we have to take into account is the background that these species that they experience in their normal cycles. So estuaries, for example, have these natural high fluctuations of pH and on a daily cycle. So what we found is that bivalves that normally live in estuaries, for example, oysters, tend to be a bit more resilient than animals that live on the continental shelf. So the species that I study is the Atlantic sea scallop, and it's a shelf species. And we found that it is more vulnerable to acidification than organisms that live closer to our shore and that experience these natural daily fluctuations. And this shows that potentially oysters could have a greater adaptive capacity, so they might be able to evolve more rapidly to keep up with acidification than a species that doesn't experience daily fluctuations.
That's really interesting. For many of us, shells are shells,
Yeah.
but what you're saying is no, that's not the case. There is presumably a chemistry, a structure
Hmm.
peculiar to different species. Could you go into that a little bit?
Sure, yeah, yeah. So as well as the shells, there's the underlying biology. So bivalves are generally osmoconformers. So they have to be able to hold their tissues at the same temperature, salinity, and pH as their environment. So animals that live close to the coasts that experience these larger fluctuations, they have different proteins and genes that allow them to cope with changing pH and changing, I think, temperature as well. Um, Normally, they only have to deal with this for a short amount of time. So that's something that a lot of scientists are looking at, is whether a short-term point exposure to ocean acidification has a different effect on bivalves than a longer-term press effect, because these kind of shock reactions that they have to sudden exposure might not be sustainable over a long period of time. Um, Another thing, as well as affecting their shells, you mentioned that we normally think of them as shells.
Yeah.
Ocean acidification can have a big effect on their tissues as well. As I mentioned, they
Yeah.
have to hold the pH of their tissues roughly the same as their environment. And when pH gets lower, that can make it harder for their enzymes to function properly, and also harder for them to do things like take up oxygen. So oxygen affinity is lower under low pH.
Wow. So there's a kind of massive effect. It's not
Hmm.
weakening of the shells, but it affects tissue as well. You
Yeah.
marine ecologist and a carbonate
Yeah, it's important to have both.
have a bunch of expertise areas in order to really penetrate this kind
Mhm.
of situation. May I ask you to talk about your research a bit because it's pretty complicated. Tell us what it is you do and how you do it. Okay.
Yeah. Yeah, so most of my research has been on Atlantic sea scallops, and as I mentioned, they live on the continental shelf. So most of their fishery takes place on George's Bank, which is this elevated bank that extends off the coast of Cape Cod and encloses the Gulf of Maine. And a large part of it also takes place in the Mid-Atlantic Bight along the coast of New Jersey. Um, so I wanted to study how they respond to ocean acidification in a couple of different ways. And first of all, I wanted to study how they might be responding in their natural habitat. A lot of ocean acidification research takes place in mesocosms and laboratory experiments. But what we see in mesocosm experiments might not be reflecting what goes on in their natural environment. So I went out, my research was funded by the Sea Scallop Research Set-Aside Award. So that's basically research that's funded by the scallop fishery. And so I went out on a scallop commercial fishing vessel and collected scallops from a number of different sites on Georges Bank. And I also measured the chemistry of the seawater at each of these sites as well, and looked for differences in both shell and tissue properties at these different sites. And I found that there was a really wide range of saturation states on Georges Bank. So yeah, so my lowest saturation state was actually just under that threshold of 1. And my highest saturation state was up around 7. So that's a really large range, which gives us some, maybe a glimmer of hope that these high saturation state sites might be robust to ocean acidification for a longer amount of time. But I did see an effect of this saturation state on both tissue and shell properties. So I found that tissue weights were lower under ocean acidification-like areas. And I also found some effects on shells. So I've got a couple images of what I found in my shells that I'd like to show you. So you should be able to see a PowerPoint just now. I'll just make that full screen. Yeah, so to look at shell properties, I used a scanning electron microscope. This is, this is the SEM that we have in the Reese lab. So I worked with Justin Reese, who is my PhD advisor. And this instrument basically fires electrons at a sample, and they get scattered differently depending on the shape of the object that they're being sent at. And then this allows us to take these really high resolution images of the shells. So this is an image of one of my shells. This is magnified 15,000 times. And each of these individual prisms is a calcium carbonate crystal. So this allows us to look at the really in-depth structure of the surface of their shells. So this was from my highest saturation state site. And you can see that, pardon?
This is like almost 7?
Yeah, this is almost 7. So this is my highest, this was the highest saturation state I found in the field. And you can see that all of those crystals, they're fairly uniform in shape. So they all have roughly the same shape. And they're also overlapping a lot. If I look at one of my lower saturation state sites, you can see that those crystals appear longer and skinnier. So we're seeing much more of it, each individual crystal. They're starting to move apart. And they're also not all the same shape. So some of them are skinny and some of them are wide. So the scallop's starting to lose control over the structure of its shell.
Wow.
Yeah, and well, let's look at the next one. So when we go to my lowest saturation state site, this was the one that was undersaturated. It's really hard to determine what an individual crystal is.
Yes.
Those crystals have really strong signs of dissolution. So this shows that this shell is actively dissolving. So I found really strong impacts in the field of seawater chemistry.
Wow.
Yeah.
When you get to that level, is it reversible? Can the
so the scallops, once they form the crystals, they don't really do much to them after that. So those crystals will stay how they are, but they're constantly growing and constantly laying down new crystals. So the fact that scallops survive at this site shows that they are able to grow under these conditions. But because there's fluctuations around the average saturation state, maybe they grow when saturation state is higher than it was when I collected this sample, for example, but don't grow as well when it drops lower. And that would be when we start to see the shells dissolving. So while they can't repair these crystals, they can grow new ones.
You go out, you collect in different areas, do this really, really fine analysis, which must be super accurate, must be tremendously helpful. And so you do this, and then you are able to determine also the interior health of the creature, right? So the tissue also, what's going on again with the tissue? And
So I'll just show you one more thing with the that I did with the shells. So I also looked at shell strength using this machine, which is an Instron. It's basically a crushing machine. So Yeah, it's just a shell, not a live scallop. So we're able to crush those shells and look at the effects of carbonate chemistry on shell strength. And I found that my shells were a lot weaker at the sites with lower saturation states. This could, for example, have an effect on their vulnerability to predation in the future. Those are all the pictures I wanted to share, but I'll stop sharing my screen. But you asked about scallop tissues.
Yes.
So I found that all of their tissues were smaller under ocean acidification. So the part of scallops that we like to eat is their abductor muscle. So if you've ever had a plate of scallops, you'll see these big chunky white meats. This is the muscle that they use to hold their shell open and closed. They actually use that muscle to aid them in swimming. If a predator approaches them, they'll swim away. And that muscle was smaller under ocean acidification. Yeah.
How about reproduction also, since they're already in a weakened condition? How about reproduction rate? Is that affected?
Yeah, that's a really good question. And it's something that is hard to get at in the field. But I did look at Gonadosomatic Index, which is the ratio of the gonad tissue to the rest of the tissue. And I found that Gonadosomatic Index was lower under my low carbonate chemistry sites.
Yes.
So this could mean a couple of different things because scallops have reproductive cycles, so they'll grow their gonads and then they will release their eggs and sperm into the water, and then they will have a very small gonad after that. So it could be that ocean acidification is affecting the rate of those cycles. So it could be that the ones from my lower saturation state sites were at an earlier stage in their development than my ones in the higher pH sites. Or it could mean that it's simply causing them to produce smaller gonads overall. And that's something that I couldn't really get at in the field, but would be really interesting to study in an experiment in the future.
Yes, and you're able to say, though, pretty clearly, this is due to acidification. There's a lot of other stuff going on in the ocean.
Yep.
Very destructive also, the toxins, plastic.
Yeah.
Are you able to separate out these kind of factors?
That's a really good question. And the answer is no, you can't do that just with a field study because, as you say, there's so much else going on in the field. There's also differences in current strength or differences in depth and food availability and so on that could be affecting my results. So to kind of take that into account, I also did a mesocosm experiment. So I brought scallops back to the lab and kept them in tanks for 120 days. And over this time, I measured their shell growth rates. I also measured their respiration rate. So one of the cool things about tank experiments in the lab is you can look a little bit more at physiology than you maybe could in the field. And then I also looked at mortality. And I found that scallops grew. Something else that you can do in a tank experiment is you can look at different factors. So I included both ocean acidification and warming in my study so that I could look at the interplay of these two climate change effects. And I found that ocean acidification caused growth to get slower. So scallops in my highest acidification treatment grew a lot less than the ones in my control treatment. But interestingly, temperature had an effect on this as well. So in my control ocean acidification treatment, scallops actually grew fastest at my highest temperature, which is kind of what we see in their natural environment as well. But under my highest ocean acidification treatment, scallops grew slowest in my highest temperature treatment. So the combination of those two stressors were just too much for the scallops. And it's really important to study those together because they're going to go occur.
Yes. That is really interesting that you're able to see how these factors do together, and they are. I take it they're kind of accelerating that the situation is actually getting worse. So there are two things. What is the effect on the fishing industry that depends on this? It must be pretty severe.
Yeah, so far we've not really seen an effect so far. And part of what my research is just now is to kind of understand how they're going to be affected in the future. So I'm collecting data on carbonate chemistry across the Northwest Atlantic shelf. to understand areas that might be more resilient to ocean acidification within the fishery and areas that might be less resilient to ocean acidification within the fishery. And although we haven't seen a large effect so far, modeling work has shown that ocean acidification is likely to have a really large impact on fisheries profits. And some different strategies for managing that could be putting more closed areas, so protecting areas where scallops are highly reproductive. So they're producing a lot of larvae and maybe seeding other areas. If we protect those areas more, then we might be able to conserve our scallop population into the future. So what I'm really trying to get at just now is the rate of ocean acidification in the field and how that's going to affect the future of the fishery.
Yes, and this acidification, is it accelerating, or is it is accelerating along with heating? How much can the ocean take?
Yeah, so that's a really good question. I'm not sure I have the answer to that off the top of my head, but it is accelerating. What acidification does is it lowers the buffering capacity of the oceans. So the ocean can buffer a certain amount of greenhouse gas input by basically, acids are positively charged, and the ocean has a lot of negatively charged ions that are naturally dissolved in seawater. So the ocean can use these ions to buffer the addition of positively charged ions. But it only has a certain amount of those negatively charged ions. So the more carbon dioxide we put into seawater, the less its buffering capacity it is, and the faster ocean acidification will occur. So I guess it's less a question of how much the ocean can take. The ocean is always going to be able to take up carbon dioxide, but more about the animals that live in it. So we really need to slow the rate of ocean acidification as much as possible to allow organisms to adapt. Because some marine organisms will be able to cope with ocean acidification. Over geologic timescales, we've seen ocean acidification in the past. So we've seen pH that gets lower, but normally it happens over a much slower rate than what is currently happening. So we know that animals can keep up if they're given the time to do so, but we really need to reduce the amount of greenhouse gases that we're putting into the ocean to slow down the rate and give them that chance.
I'm so glad you brought up that rate issue because people sometimes doubt the situation. Need to realize that when we're talking about eons on which these changes take place, that's totally different from our situation now.
Exactly. Yeah.
Thank you for bringing that up. But what's your prognosis as far as, well, you're concentrating on one species, but this kind of life, the shellfish, can't migrate very easily. Get on a bus and go somewhere else. So I understand that certain coastal regions are really quite affected, but what's your prognosis for the near future? Let's hope that we'll turn a corner in policy, but it has to be international, it has to be quick. What's your idea about what will happen in, say, 10 years?
Yeah, I think it's difficult. It really depends on the system that you're looking at. I think, unfortunately, some carbonates like corals have reached a point where if we don't do something now, they're just not going to recover because they're also dealing with warming. With bivalves, we've found that bivalves are so strange. They'll grow in sites where you would never expect them to be able to grow sometimes. So I think that if we are careful and if we make sure that we're not overfishing our species and that we're also slowing the rate of greenhouse gas input into the oceans, the bivalves will be okay, but it's going to take a lot of work to do that. So that's going to take a combination, as you say, of policy, human activities, human behavior, and also making sure that we're fishing in the right place and not reducing bycatch and so on.
Right. The other part of that you brought up that was so interesting was the issue of runoff. So you think of the Gulf of Mexico, these Gulf areas that get so much of that runoff, and then the effect of that with then do they have to change policies some way also?
Yeah, absolutely. And that's something that we even see up here in Massachusetts. We still see a lot of runoff into our coastal waters. And that's something that can affect not Atlantic sea scallops as much, but more of our coastal species, for example, oysters, bay scallops, mussels. This is something that having that extra stressor that they're having to deal with. So as well as causing, really quick acidification, this also causes a loss of oxygen as well. So they're dealing with this stressor causes, is harmful to marine organisms in a number of different ways. So that's definitely something, and that's something that's much easier to reduce, right, than, suddenly stopping greenhouse gas production. If we put efforts into preventing runoff, that could have a really big effect. Yeah.
Yes. So you have your work cut out for you. Just one more quick thing about your particular training, because you have the chemistry background along with the ecology background. Is that typical today that people looking at marine life need the two?
I think that not necessarily one person, for example. I think that it's definitely becoming more common to be interdisciplinary, but also to collaborate with other disciplines. So, for example, I have very poor genetics knowledge, but I collaborate with geneticists because they have that expertise to be able to predict rates of evolution and so forth. And I think that, it's going to take scientists from a lot of different disciplines working together. Also, modeling skills are super important. So I'm collaborating with modelers because that's something that's really provides really compelling evidence for policymakers. If you look at, the monetary cost of acidification, for example, that's not something that I have expertise in, but something that someone else I could work with would have. Yeah.
That is, I think it must be very exciting work in addition to being demanding and maybe a little depressing if you can't do anything about it. Is your scientific community able to put pressure on governments, on institutions to help make people aware that they have to do something at the policy level?
Yeah, that's difficult. Scientists, it's part of our ethos that we have to remain neutral and just kind of, do the science and, get the facts out there and get our data, make our data as publicly available as possible. And that's why we do things like this, so that we can get the information out to other people and other groups who can maybe put pressure on governments. We just try and kind of give tell the truth and say what's happening.
Yes, I understand. I understand. It's very difficult for the science community, and yet they need to scream that this is a little bit urgent here.
Yeah.
I think it's just a very interesting field, a wonderful field, and I wish you the absolute best luck with your work. I'm sure it is a wonderful place to be at Woods Hole.
Yeah.
Just exactly what you'd like to be doing. And I thank you so much for joining us today and giving this public really good information. Thank you.
Yeah, thank you very much. Thanks so much for having me.